Welcome back to the deep dive. Today, we are undertaking, well, a really critical mission, actually, taking a comprehensive deep dive into irreversible cell damage. We're moving past those subtle, recoverable stresses.
Reversible stuff.
Exactly. And focusing entirely on necrosis, this uncontrolled pathologic cell death. It often marks a major clinical event.
It really does. It's messy, it's destructive, and it's basically the body's ultimate signal that something is irreparably injured.
Yeah, like the cell just falls apart.
Pretty much. Necrosis is essentially the cellular equivalent of a complete structural collapse. When damage gets severe enough, the cell just can't adapt. It fails spectacularly sometimes.
So we needed to find that line, right? The point of no return you mentioned.
Precisely. That line in the sand where a cell goes from a state where, it could bounce back, that's reversible injury, to irreversible injury, which inevitably leads to this catastrophic process we call necrosis.
Okay, let's unpack that, because cells swell a bit or have mitochondrial hiccups all the time under stress. But what are the specific failures you mentioned, too, that really define that threshold, that difference between just a bad day for the cell and, total irreversible death?
Yeah, the sources are quite consistent here. They identify two key interrelated phenomena that really seal the cell's fate. They signify total system failure.
Total system failure, okay.
And it mainly revolves around energy and structural integrity. The first one is the permanent, irreversible inability to restore mitochondrial function.
So the power grid is just gone for good. No fixing it.
That's a great analogy. If the mitochondria, the cell's powerhouses, are damaged beyond repair, they simply cannot synthesize ATP anymore, even if oxygen and nutrients suddenly flood back in.
Even if the original problem is fixed.
Right. It's too late. Without ATP, every single energy-dependent function just grinds to a halt. The pumps maintaining osmotic balance fail, protein synthesis machinery stops. Yeah. The cell just can't generate the bare minimum power needed to live.
And without those pumps keeping the balance, water rushes in, the cell swells up, which I guess connects directly to the second irreversible marker.
It does, absolutely. The second marker is a profound, widespread disturbance in all cellular membranes, especially the plasma membrane, the outer wall, and, crucially, the lysosomal membranes.
Ah, the lysosomes. Those are the cell's sort of recycling centers?
Yeah.
But with dangerous stuff inside.
Exactly. Powerful recycling and waste disposal units packed with highly aggressive hydrolytic enzymes designed to break down cellular junk or invaders.
But if their own membranes break,
then disaster strikes. When the plasma membrane and, critically, those lysosomal membranes rupture due to this irreparable damage, those digestive enzymes spill out into the cytoplasm.
Oh, so instead of digesting waste, they turn on the cell itself.
It's autodigestion, self-feeding. The enzymatic dissolution is really a defining biochemical mechanism of necrosis.
Which brings us right back to the definition then.
Right. Necrosis is, by definition, a pathologic process. It's never normal, never physiologic, resulting from overwhelming, severe damage. And it's characterized by that uncontrolled cell swelling, membrane rupture, and breakdown.
So what are the big triggers, the overwhelming insults that push a cell past this tipping point?
The triggers are usually severe, often acute. Loss of oxygen supply, ischemia is probably the most common cause. Think heart attack, stroke.
Right. Lack of blood flow.
Exactly. Other key triggers include direct physical injury, like major trauma, thermal injury, severe burns, microbial toxins that directly poison or damage cell structures.
Chemicals too, I assume.
Oh, yes. Chemical or physical injury, certainly. And there's an interesting specific situation, acute pancreatitis.
Ah, yeah, where the pancreas kind of digests itself.
In a way, yes. Uncontrolled activation of digestive enzymes like proteases and lipases causes them to leak out and start digesting tissues outside the gut. It leads to widespread necrosis in the pancreas and surrounding abdominal tissues. Really nasty stuff.
Okay, so to really get a handle on this messy, accidental necrosis, we need to contrast it sharply with the other main type of cell death, apoptosis.
Absolutely crucial distinction.
The so-called programmed cell death. Clinically, understanding this difference is huge for diagnosis, for treatment. So let's break down the key distinctions our source material highlights as a table, right?
Yes, Table 2.1 in the source lays it out very clearly. The distinction is fundamentally about control versus chaos. Apoptosis is regulated, neat, deliberate. Necrosis is chaotic, messy, essentially accidental cell demolition.
Okay, where do we start? Cell size.
Good place to start. In necrosis, remember those failing ATP pumps? Water rushes in, so the cell enlarges, it swells up. sometimes quite dramatically.
But apoptosis.
Apoptosis involves cell shrinkage. The cell condenses down.
Swelling versus shrinkage. That's a big visual clue for a pathologist looking at tissue.
A major one. But perhaps the single most important difference, mechanistically and clinically, is membrane integrity. In necrosis, the plasma membrane is disrupted. It bursts open, it lyses, spilling all the cellular contents out into the surrounding tissue.
Total breakdown of the outer wall.
Total breakdown. Apoptosis, by stark contrast, is characterized by an intact plasma membrane throughout the process. Even as the cell is dismantling itself internally, it neatly packages its contents into small membrane-bound sacs.
Those are the apoptotic bodies.
Exactly, apoptotic bodies, which are then cleaned up quietly.
And that difference in membrane integrity, bursts open versus neatly packaged, explains why necrosis is so loud and disruptive, creating that massive alarm we talked about earlier.
Precisely. Because necrotic cell contents leak out indiscriminately, they act as powerful danger signals to the immune system. This causes a frequent, often severe inflammatory reaction right next to the dying cells.
The body sends in the troops, macrophages, neutrophils, to clean up the explosion and deal with the debris.
Right, it's a 5-alarm fire. Apoptosis, though. It's a silent, clean removal. Phagocytes recognize specific signals on the surface of those intact apoptotic bodies and just engulf them without ever seeing the internal contents. So critically, apoptosis induces no inflammation.
And their roles are different too, right? Necrosis is always bad news.
Necrosis is always pathologic. It's the endpoint of irreversible injury. Apoptosis, on the other hand, is often a necessary physiologic process, getting rid of unwanted cells during embryonic development, eliminating old cells, immune system regulation. That's often apoptosis.
Okay, so this contrast really matters for you, the listener, because when your doctor is looking for signs of tissue injury, they're often measuring the fallout from that necrotic bursts, the inflammation, the specific proteins that have leaked out.
That's exactly right.
All right, let's dive deeper into the mechanics then. We've established the membranes rupture, enzymes leak out. But what exactly are those enzymes doing inside the collapsing cell, and what does it actually look like as it breaks down?
So, after that irreparable membrane damage, the cell's fate is sealed by really two main biochemical events happening simultaneously: enzymatic digestion and protein denaturation.
Okay.
The lysosomal enzymes, now free in the cytoplasm, just start breaking everything down. They digest structural proteins, the DNA and the nucleus, the organelles like mitochondria. It's an internal demolition.
Total chaos.
Total chaos. And at the same time, the dying cell often becomes quite acidic, which helps to denature many of the cytoplasmic proteins. They lose their shape, their function. So the cell becomes this sort of structureless, fragmented, Legolized collection of debris.
And when all that internal stuff leaks out, it's not just passive garbage. You mentioned danger signals.
Definitely not passive. These leaked contents become active danger signals. We call them damage-associated molecular patterns, or DAMPs.
DAMPs, okay.
These are molecules that are normally hidden safely inside the cell. Things like ATP, DNA fragments, certain proteins.
Wait, ATP, the energy molecule. How is that a danger signal?
Yeah.
I thought it was good.
Inside the cell, it's essential energy currency. But it should never be floating around in large amounts outside the cell.
Okay. Context matters.
Huge context matters. When the cell membrane ruptures during necrosis, massive amounts of ATP leak out. This extracellular ATP binds to specific receptors called purinergic receptors on nearby immune cells like macrophages and dendritic cells.
And that binding triggers them.
It triggers their activation, yes. It's like the cell screaming, "Massive energy failure, catastrophic breach here." The
Yeah.
energy molecule itself becomes a powerful alarm bell.
Wow. Okay. And the other DAMP you mentioned was uric acid, like in gout.
Yes, the very same molecule. Uric acid is a breakdown product of DNA, specifically purines. When cells rupture and release their nuclear contents, uric acid levels rise locally.
And that also triggers inflammation.
It does. Extracellular uric acid, especially if it forms crystals, can activate immune cells through various pathways, including things called toll-like receptors and the inflammasome complex, which we might touch on later.
Okay. So these DAMPs, ATP, uric acid, probably others too, leak out.
Right. And they trigger receptors on immune cells. This initiates phagocytosis, the cleanup crew arrives, but critically, it also induces the production and release of a whole host of inflammatory cytokines.
Like messengers calling in more troops and coordinating the response?
Exactly. Messengers like TNF, interleukin-1, these sustain and amplify the acute inflammation we see clinically around necrotic tissue. It's a positive feedback loop. Leakage causes damps. Damps cause inflammation. Inflammation brings in cells that might cause more damage or cleanup. It's a whole cascade.
And here is where we connect this microscopic chaos directly to the clinic, to the bedside, that leakage you just described, that messy spilling of internal contents. That's precisely what allows doctors to diagnose acute, ongoing tissue damage.
It's absolutely fundamental to modern clinical diagnosis for so many acute conditions. Think about it. Different tissues, heart, liver, skeletal muscle, pancreas, they all contain unique proteins, specific enzymes that do their specialized jobs.
Right. They're tissue-specific markers.
Exactly. When those cells die necrotically and their membranes rupture, these unique internal proteins leak out into the bloodstream, and we can measure them.
So we're essentially measuring the cellular debris in the blood.
We are measuring the direct biochemical aftermath of necrosis, often in real time. It provides an incredibly powerful diagnostic window.
Okay, let's use the prime example, the one everyone knows from ER shows. Cardiac troponin for diagnosing a heart attack.
Ugh.
Why is that molecule so good, so sensitive, so specific?
Troponin is a fantastic example. It's actually a complex of 3 proteins, troponin C, I, and T, that regulate muscle contraction. Now, the critical thing is that the specific isoforms of troponin I and troponin T found in heart muscle, we call them TTNI and CTNT, are structurally unique.
Different from the troponin in, say, my bicep.
Very different. And crucially, they are virtually undetectable in the blood of a healthy person. They should stay inside the heart muscle cells.
So if they show up in a blood test,
it means heart muscle cells have died. Specifically, they've undergone necrosis, their membranes have ruptured, and these proteins have leaked out into the circulation. It's a direct indicator of myocardial infarction heart attack.
and the timing is key here, right?
Yeah.
How quickly does this signal appear after the damage starts? That seems vital for treatment.
It's remarkably fast and sensitive. We can start detecting elevated levels of cardiac troponins in the blood serum as early as 2 to 3 hours after the onset of myocardial cell necrosis.
2 to 3 hours, that's incredibly early.
It is. This is often well before a patient might even present with classic symptoms, or certainly long before any definitive changes would be visible on an EKG, or if you were somehow looking at the heart tissue itself.
And it stays elevated for a while.
Yes, that's also important. Depending on the specific troponin and the extent of damage, they can remain elevated for days, even up to 2 weeks in the case of CTNT. This gives us both an early diagnostic window and a way to track the injury over time.
That sensitivity and specificity, appearing so early, it allows clinicians to diagnose much faster and start aggressive treatment, like opening up blocked arteries, way sooner than relying on older, less specific markers or just symptoms alone.
Absolutely. Troponin testing revolutionized cardiology. The cell's explosion, its necrotic death, literally gives us a biochemical warning within just a couple of hours.
And this principle applies elsewhere too, right? Not just the heart.
Oh, definitely. The principle is the same across the body. If we suspect liver damage, say from hepatitis or a drug reaction, we measure liver-specific enzymes like alanine aminotransferase, ALT, and aspartate aminotransferase, AST.
The liver function tests.
Right, those transaminases are highly concentrated inside hepatocytes, liver cells. If they leak out, it signals liver cell necrosis. Similarly, if we suspect damage to the bile ducts, we look for specific isoforms of alkaline phosphatase. Measuring these leaked tissue-specific proteins is the clinical confirmation that necrosis has happened or is happening.
Okay, so we've covered the clinical detection through leakage. Now, let's put the blood test aside and actually look through the microscope, because the dying cell itself tells a story visually. What are the key clues a pathologist looks for when examining necrotic tissues stained with H&E? That's hematoxylin and eosin, the standard pink and blue stain, right?
Yes, H&E is the workhorse stain in pathology. Hematoxylin stains nuclei and RNA blue or purple, basophilic, and eosin stains proteins, especially in the cytoplasm, pink or red, eosinophilic. A necrotic cell undergoes really striking visual changes.
What's the first thing you notice?
Often, the first and most obvious change in the cytoplasm is increased eosinophilia. The cytoplasm appears much redder, much pinker than its healthy neighbors.
Brighter pink. Okay, why does it suddenly turn so red?
It's actually for 2 main reasons. First, the cell is actively degrading its cytoplasmic RNA. RNA normally binds the blue hematoxylin stain, so as the RNA disappears, the cytoplasm loses its bluish tinge.
Less blue. Got it.
Second, and probably more significantly, those structural proteins inside the cytoplasm are denaturing. Remember we talked about denaturation?
Yeah, losing their shape.
Right. When proteins denature and unfold, they expose more sites that can bind to the eosin dye. So the denatured proteins soak up more of the pink red eosin, lose the blue, gain more red. The net effect is a much brighter pink or red appearance.
It looks almost glassy or homogeneous sometimes.
Exactly. It often takes on a glassy, homogeneous look, partly because the cell also loses its glycogen particles, which normally give the cytoplasm a sort of granular texture. Then, as the process continues and those leaked lysosomal enzymes start digesting the internal organelles, the cytoplasm can become vacuolated. It develops empty-looking spaces, giving it a moth-eaten or almost Swiss cheese appearance under high power.
Moth-eaten cytoplasm.
Okay.
Anything else in the cytoplasm?
Yes. Another interesting feature you can sometimes see are myelin figures. These look like large, sort of whorled-laminated structures.
Whorls, like fingerprints.
A bit, yeah. They're essentially aggregates of damaged cellular membranes, mostly phospholipids, that have precipitated out. They're not always prominent, but they are a sign of severe membrane damage. And they're important because these phospholipid aggregates can later attract calcium salts.
Leading to calcification.
Leading to what we call dystrophic calcification, yes. Calcium depositing in dead or dying tissue. Those myelin figures can be a starting point for that.
Okay, so the cytoplasm gets redder, glassier, maybe moth-eaten, and can have these myelin figures. But the nucleus, that's the command center with the DNA. Surely it undergoes some dramatic changes, too.
Yeah.
The source mentioned 3 specific fates.
Oh, absolutely. The nuclear changes are perhaps the most definitive signs of irreversible cell death, of necrosis. They follow a fairly predictable sequence, representing the progressive breakdown of DNA and chromatin. The source describes 3 distinct patterns: karyolysis, pinosis, and karyorexis.
Okay, let's take them one by one. Karyolysis. Lysis means breaking down.
Exactly. Karyolysis literally means dissolution of the nucleus. In this stage, the blue color, the basophilia of the chromatin, starts to fade. This is because enzymes, specifically endonucleases, are starting to degrade the DNA.
So the nucleus looks pale, washed out.
Precisely. It becomes ghostly, indistinct. The blue color fades away over time.
Okay, so first it fades.
Yeah.
That's karyolysis. What's next? Pinosis. That sounds different.
Pinosis is almost the opposite visually. Here, the nucleus shrinks dramatically in size, and as it shrinks, the chromatin condenses down into a small, incredibly dense, solid-looking mass. Because it's so condensed, it stains very intensely dark blue or almost black with hematoxylin.
So it goes from faded and pale, karyolysis, to small, dark, and dense pinosis, like a little ink dot.
A perfect description. A small, dense, hyperbasophilic ball, that's pinosis.
And the third stage, karyorexis. That sounds like breaking.
Rexis means rupture or fragmentation. Karyorexis is the stage where that dense pinotic nucleus breaks apart. It fragments into multiple small, dark, dusty-looking pieces of chromatin scattered usually within the dying cell's cytoplasm.
So it shatters, basically.
It shatters. And then, usually within about 1 to 2 days, those nuclear fragments are completely degraded and disappear entirely. What you're left with is just the enucleated, no nucleus, eosinophilic red ghost of the cell cytoplasm. Seeing any of these nuclear changes, karyolysis, pinosis, or karyorexis, confirms you're looking at irreversible necrotic cell death.
Wow. Fading, shrinking, shattering, a pretty definitive end for the nucleus.
Absolutely.
Now, when necrosis doesn't just affect single cells, but large groups of cells, entire tissues, the overall pattern of tissue destruction can look different depending on the cause and the tissue type. Our sources describe 6 classic morphological patterns, right?
That's correct. These patterns are really important because seeing one of them grossly with the naked eye or microscopically gives pathologists and clinicians huge clues about the underlying disease process.
Okay, let's go through them. First up, probably the most common one, coagulative necrosis. What defines that?
Coagulative necrosis is defined by something quite remarkable. The basic architecture, the outline of the dead tissue, is actually preserved for a period, often several days.
Preserved, even though the cells are dead. How does that work?
Well, the tissue usually feels firm, sometimes a bit rubbery. Think of, well, cooking an egg white. It goes from liquid to firm, right? Coagulating.
Okay, so the proteins solidify. But why doesn't it just break down? Why don't those lysosomal enzymes just digest everything immediately?
That's the key mechanistic insight for coagulative necrosis. The injury, which is typically severe ischemia, a sudden loss of blood supply.
Like in a heart attack or kidney infarct.
Exactly. That severe ischemia not only kills the cells by starving them of oxygen, but it also denatures both the structural proteins and, crucially, the cell's own degradative enzymes, the lysosomal hydrolases.
So the demolition crew gets knocked out along with the building residents.
Perfect analogy. Because the enzymes are denatured and inactivated, they can't perform autolysis, they can't digest the cell from within. So the cell dies, the nucleus undergoes those changes we discussed, pinosis, karyolysis, but the basic outline of the cell and the tissue framework persists for a while.
So a pathologist can still recognize the structure?
Yes. They can look at an area of coagulative necrosis in the kidney, for instance, and still see the ghostly outlines of the renal tubules or glomeruli. Or in the heart, they see the outlines of cardiac muscle fibers. The cells are dead, eosinophilic cytoplasm, no nuclei. But the blueprint remains visible for days until inflammatory cells eventually arrive to clear the debris.
And the classic example is an infarct, that area of dead tissue from ischemia in solid organs.
Yes, infarcts in the heart, kidney, spleen. They typically appear as wedge-shaped, pale or yellowish areas of coagulative necrosis. The one major exception, interestingly, is the brain.
The brain does something different. We'll get to that.
Okay.
So coagulative, structure preserved because enzymes are knocked out. Now, let's contrast that sharply with liquefactive necrosis, the name suggests.
Liquefaction, turning into liquid. This is the complete opposite of coagulative. Here, the dead cells are completely digested, transforming the tissue into a viscous liquid mass. There's no preservation of architecture.
Total breakdown. What causes that?
The classic cause is focal bacterial or sometimes fungal infections.
What?
Why? Because these microbes are powerful attractants for neutrophils, those white blood cells.
The inflammatory cells.
Right. And neutrophils are basically bags of potent hydrolytic enzymes. When huge numbers of them swarm into an infected area and die, they release all those enzymes.
So the enzymes come from the inflammatory response, not just the dead tissue cells.
Exactly. It's the combination of enzymes from the dying neutrophils and enzymes perhaps produced by the bacteria themselves that leads to this rapid, complete digestion of the necrotic tissue.
And that resulting liquid, that's what we call pus.
Precisely. The creamy, yellowish, viscous material formed in liquefactive necrosis due to infection is, pathologically speaking, pus. It's basically dead and dying neutrophils, liquefied tissue debris, and often the microbes themselves.
Okay, infections cause liquefactive necrosis, but you mentioned the brain was an exception for coagulative. Does it do liquefactive instead?
It does. This is a crucial point. Hypoxic injury in the central nervous system, like from a stroke causing brain infarction, uniquely results in liquefactive necrosis, even without infection.
Why does the brain liquefy when the heart coagulates?
The thinking is that the brain has very little supportive connective tissue stroma compared to other organs, and it's very rich in lipids and, of course, water. It also seems to have plenty of its own hydrolytic enzymes in glial cells. So when brain cells die from hypoxia, they tend to undergo rapid dissolution, leaving behind a fluid-filled cavity or cyst once the debris is cleared.
Fascinating. Okay, pattern 3, gangrenous necrosis. The name itself sounds pretty grim.
Gangrene is a term used more in a clinical context, often by surgeons, rather than being a distinct microscopic pattern like coagulative or liquefactive. It's usually applied to a limb, typically a lower leg or foot, that has lost its blood supply and undergone extensive necrosis involving multiple tissue layers, skin, muscle, connective tissue.
So it's basically widespread necrosis of a limb due to ischemia.
Correct. Now, clinically, we often subdivide it. If the main underlying process is just the ischemia, leading primarily to coagulative necrosis of the tissues, it's termed dry gangrene. The limb becomes dry, shriveled, dark, blackish brown.
Like mummified.
Somewhat, yes. But the danger comes if that dead tissue gets infected.
So infection complicates things again.
Big time. If a bacterial infection is superimposed on the ischemic necrotic tissue, then the actions of bacterial enzymes and the resulting inflammatory response with neutrophils releasing their enzymes add an element of liquefactive necrosis on top of the coagulative background.
Making it wet.
Exactly. This transforms it into wet gangrene. The tissue becomes moist, swollen, often foul-smelling due to bacterial decomposition, and the liquefaction causes much more rapid tissue destruction. Wet gangrene is a surgical emergency.
Okay, dry is mostly coagulative. Wet has added liquefactive due to infection. Got it. Pattern 4, caseous necrosis. This one's defined by its appearance, right? Cheese-like.
Yes, the term caseous comes from casein, the protein in cheese. Grossly, the necrotic area looks soft, white, or yellowish, friable, crumbly, like cottage cheese or feta cheese.
Okay. And what condition is most famously associated with this?
This pattern is encountered most often, almost characteristically, in foci of tuberculous infection, tuberculosis.
TB. So if a pathologist sees caseous necrosis, they immediately think of TB.
It's very high on the list, yes. Microscopically, caseous necrosis is also quite distinct. The necrotic area is completely structureless. You can't make out any cell outlines at all. It appears as amorphous, granular, pinkish debris with H&E stain.
Total obliteration of the tissue architecture.
Complete obliteration. And crucially, this caseous debris is typically surrounded by a very specific type of inflammatory border, a collection of activated macrophages, called epithelioid cells, and lymphocytes, often with giant cells. This entire structure, the central caseous necrosis surrounded by the inflammatory cells, is called a granuloma.
The granuloma tries to wall off the infection and the necrosis.
Precisely. Finding caseous necrosis within a granuloma is a hallmark of tuberculosis, although some fungal infections can occasionally cause a similar picture.
Okay, cheese-like center and granuloma, think TB. Next, fat necrosis. Sounds specific to fat tissue.
It is. Fat necrosis refers to focal areas of fat destruction. It has a very specific mechanism, typically resulting from the release of activated pancreatic enzymes, particularly lipases, into fatty tissue.
Pancreatic enzymes again, like in pancreatitis.
Exactly. The classic example is acute pancreatitis. When the pancreas gets severely inflamed, those powerful digestive enzymes leak out of the damaged pancreatic acinar cells and escape into the surrounding retroperitoneal fat and even the abdominal cavity.
And the lipases specifically attack the fat cells.
Yes. Lipases break down the triglycerides stored within fat cells, adipocytes. This hydrolysis releases fatty acids.
Okay.
Now, these released fatty acids aren't soluble. They immediately react with calcium ions, which are present in the tissue fluids and circulation. This chemical reaction forms calcium soaps.
Soap, like actual soap.
Chemically, yes. It's the process of saponification. Fatty acids plus calcium or other alkali metals make soap. These calcium soaps are insoluble and precipitate out in the damaged fatty tissue.
And what do these calcium soaps look like?
Grossly, they appear as chalky white, firm, sometimes gritty deposits scattered throughout the affected fatty tissue. Finding these telltale white flecks of fat saponification during surgery or autopsy is a classic sign of past or present acute pancreatitis. Microscopically, you see the necrotic fat cells, often with shadowy outlines surrounded by inflammation and these basophilic, blue-staining calcium deposits.
Chalky white spots from fat turning into soap.
Got it.
Finally, the sixth pattern, fibrinoid necrosis. Sounds like it involves fibrin.
It does, or at least it looks like fibrin. Fibrinoid necrosis is a special type of necrosis, usually seen only in the walls of blood vessels.
Ah, specific to vessels. What causes it?
It's typically associated with immune-mediated vascular damage. Think conditions like vasculitis syndromes, where immune complexes, antibody clumps, get deposited in artery walls. It can also be seen in severe malignant hypertension, where extreme blood pressure physically damages the vessel wall.
Okay, immune complexes or severe hypertension damage the vessel wall. What happens then?
The damaged vessel wall becomes more permeable. Plasma proteins, including fibrinogen, which converts to fibrin, leak out from the blood into the vessel wall. These leaked proteins, along with the deposited immune complexes and necrotic smooth muscle debris, mix together.
And how does that look under the microscope with H&E?
This mixture stains intensely bright pink or red with eosin, and it looks amorphous, glassy, and very much like fibrin, hence the name fibrinoid. So pathologists see this bright pink, smudgy deposit within the vessel wall, obscuring the normal structure. It's a clear sign of severe vascular injury, often immune-related.
Bright pink staining in a vessel wall. Fibrinoid. Okay, those are the 6 classic patterns.
Yeah.
Coagulative, liquefactive, gangrenous, caseous, fat, and fibrinoid. Each tells a different story.
They really do. They provide a vital link between the microscopic cell death and the macroscopic disease process.
Now, everything we've discussed so far fits the classical view of necrosis as this uncontrolled, accidental, messy process. Just cellular collapse due to overwhelming injury.
That was certainly the dogma for a very long time. Necrosis was the accident, apoptosis was the programmed suicide, clear distinction.
But research in the last, decade or two has really blurred those lines.
Fundamentally shifted the view, yes. We now recognize several distinct forms of cell death that absolutely look like necrosis. Morphologically, you get the cell swelling, the membrane rupture, the inflammation, all the hallmarks of classical necrosis.
Okay.
But crucially, their initiation and execution are tightly regulated by specific genetically encoded molecular pathways, just like apoptosis.
So it's a regulated explosion, programmed messiness.
That's a great way to put it. This discovery that necrosis isn't always accidental, that it can be programmed, has opened up entirely new ways of thinking about disease and potential therapies.
This sounds revolutionary. Let's start with the big one, necroptosis. What makes it this hybrid programmed necrosis?
Necroptosis perfectly embodies this hybrid concept. Morphologically, under the microscope, it looks like necrosis. Cell swelling, organelle swelling, eventual plasma membrane rupture, release of damps, and subsequent inflammation. Classic necrotic features.
Just like necrosis,
Huh.
but mechanistically.
Mechanistically, it's regulated. It's triggered by specific signaling pathways, often initiated by the same sorts of signals that can trigger apoptosis, like activation of death receptors.
So if it's regulated like apoptosis, why isn't it just called a type of apoptosis? What's the absolute key molecular difference?
The single most important defining feature is that necroptosis is completely caspase independent.
No caspases.
Right.
But aren't caspases the executioner enzymes for apoptosis?
Exactly. Classical apoptosis relies entirely on a cascade of enzymes called caspases, particularly the executioner caspases 3 and 7, to dismantle the cell neatly. Necroptosis occurs specifically when the caspase cascade is blocked or inhibited, perhaps by viral proteins or certain cellular stresses, yet the cell still receives a death signal.
So the cell gets the death signal, the normal suicide pathway, apoptosis, is blocked.
Yeah.
It finds another way to die, a messier way.
Precisely. It activates a backup caspase-independent execution pathway that leads to this necrotic morphology.
Okay, so how does it pull off this self-destruction without caspases? What's the molecular chain of command? Who are the executioners in necroptosis?
The signaling often starts, as I mentioned, with death receptors like TNFR1, the receptor for tumor necrosis factor. When TNFR1 is activated and if castases, specifically caspase 8, are inhibited,
The backup plan kicks in.
the backup plan kicks in. The receptor complex recruits two crucial protein kinases, RIPK1 and RIPK3. That's receptor interacting protein kinase 1 and 3.
RIP kinases, okay.
These two kinases are the central regulators of necroptosis. They interact, phosphorylate each other, and form a signaling complex sometimes called the necrosome.
The necrosome, that sounds ominous.
It is for the cell. The key downstream target, the final executioner molecule activated by the necrosome, specifically by RIPK3, is a protein called MLKL. That's mixed lineage kinase domain-like protein.
MLKL. And what does activated MLKL do to actually cause the cell to blow up?
RIPK3 phosphorylates MLKL. This phosphorylation is the trigger. It causes MLKL monomers to change shape and assemble into larger complexes, oligomers.
Okay, they team up.
They team up, and these MLKL oligomers then rapidly move to and insert themselves into the plasma membrane and also mitochondrial membranes.
They punch holes in the membranes.
Essentially, yes. They form pores or channels that disrupt the membrane's integrity. Water rushes in uncontrollably, ions leak out, the cell swells rapidly, and eventually the plasma membrane ruptures. Boom! Necrotic death, release of damps, inflammation.
Wow, so RIPK1 and RIPK3 activate MLKL, and MLKL is the protein that physically destroys the membrane. That's incredibly specific.
It is. And identifying the specific RIPK1, RIPK3, MLKL pathway has been huge.
Because if you can block it,
You could potentially prevent that necrotic cell death, even if the initial trigger occurred. This pathway isn't just some obscure biological curiosity. It's now known to be a major contributor to cell death in many important human diseases.
like what? Where does necroptosis play a role?
It's implicated in the massive cell death seen in acute pancreatitis, certain types of liver disease like steatohepatitis, fatty liver disease, and importantly, in ischemia reperfusion injury.
Ischemia reperfusion. That's when blood flow is cut off, then restored, right? Like after a heart attack or stroke is treated.
Exactly. Restoring blood flow is essential, but the reperfusion itself can trigger a wave of necroptosis in the cells that survive the initial ischemia, paradoxically worsening the damage. Necroptosis is also implicated in some neurodegenerative diseases like Parkinson's disease, potentially contributing to the loss of neurons.
So developing drugs that inhibit RIPK1 or RIPK3, that could be huge therapeutically.
It's a major focus of drug development right now. Necrostatin 1, for example, is an inhibitor of RIPK1 being studied.
Okay, necroptosis. Programmed, caspase-independent, RIPK13 MLKL-driven membrane rupture. Now, what about pyroptosis? Pyro, that means fire or fever.
Yes, pyro refers to fever. Pyroptosis is another form of regulated programmed cell death, but it's distinguished by being inherently and intensely inflammatory. Its defining feature is the release of highly pro-inflammatory cytokines, especially interleukin-1 beta, IL-1, which is a major fever-inducing molecule.
So it's a cell death that actively signals fever and inflammation. What triggers this fiery death?
Pyroptosis is typically triggered by intracellular pathogens, bacteria, or viruses that get inside cells, or by certain endogenous danger signals. These triggers are recognized by specific sensor proteins within the cell's cytoplasm.
Okay.
This recognition leads to the assembly of a large multi-protein complex called the inflammasome. The inflammasome is the central molecular machine driving pyroptosis.
Inflammasome. Sounds important. What does it do once it's assembled?
The assembled inflammasome acts as a platform to activate specific inflammatory caspases, primarily caspase 1, and sometimes caspase 11 in mice, or caspase 45 in humans.
Caspases again, but these are inflammatory caspases like caspase 1, not the executioner caspases like caspase 3 used in apoptosis.
Yeah.
Different family members, different jobs.
Okay, different caspases. So what does activated caspase 1 do in pyroptosis? Activated caspase 1 has two main jobs. First, it finds the inactive precursor forms of those key inflammatory cytokines, pro-IL-1 and pro-IL-18, and cleaves them into their active mature forms. These active cytokines are then released from the dying cell to sound the alarm.
Job 1.
Right.
Release the inflammatory flares. What's job 2?
Job 2 is to execute the cell death itself. Caspase 1 cleaves another protein called gastromin D.
Gastromin D.
Yes. When gastromin D is cleaved by caspase 1, its N-terminal fragment oligomerizes and, much like MLKL in necroptosis, inserts into the plasma membrane, forming large pores.
Pores again, leading to rupture.
Leading to osmotic lysis, cell swelling, membrane rupture, and release of the cellular contents, including those activated cytokines IL-1 and IL-18. So the cell dies and simultaneously releases potent inflammatory signals.
It's like a suicide bomber cell, taking itself out while releasing signals to rally the immune system against the infection.
That's a very apt description. It's a highly effective, albeit destructive, host defense mechanism against intracellular pathogens. Sacrifice the infected cell, but make sure the immune system knows about it loudly.
Pyroptosis. Inflammatory, inflammasome caspase 1 driven, gastromin D pores, cytokine release.
Got it.
Now, the last one mentioned is ferroptosis. This sounds different again. Ferro, iron.
Exactly. Ferroptosis is a unique form of regulated cell death characterized by its absolute dependence on iron and by the accumulation of lethal levels of lipid peroxidation.
Lipid peroxidation.
Yeah.
Like fats going rancid, rusting membranes.
That's a great way to think about it. It's oxidative damage specifically to the lipids within cell membranes. Ferroptosis is triggered when there's an imbalance, either too much iron floating around or excessive reactive oxygen species, ROS, overwhelming the cell's antioxidant defense systems.
We hear a lot about antioxidants being good. What specific defense system fails in ferroptosis?
The key defense system that fails or is inhibited in ferroptosis is the one dependent on glutathione, specifically an enzyme called glutathione peroxidase 4, GPX4.
GPX4, what does it normally do?
GPX4's main job is to detoxify those dangerous lipid peroxides, using glutathione as a cofactor. It neutralizes them, preventing them from accumulating and chain reacting within the membranes.
So in ferroptosis, either there's too much iron generating too many peroxides, or the GPX4 enzyme itself isn't working properly.
Correct. Iron, particularly free ferrous iron, Fu2+, acts as a catalyst in reactions that generate ROS and directly promote lipid peroxidation, like the Fenton reaction. If GPX4 activity is low, due to lack of glutathione or direct inhibition, it can't keep up with the damage. The lipid peroxides build up uncontrollably.
And that buildup physically destroys the membranes.
It does. The peroxidation causes extensive damage to the structure and fluidity of cell membranes, especially the plasma membrane, leading to increased permeability, loss of integrity, and ultimately cell death that morphologically resembles necrosis, swelling, rupture.
So it looks like necrosis, but the underlying cause is this specific biochemical pathway of iron-dependent lipid peroxidation.
Precisely. It's a biochemically distinct death pathway.
Is there anything unique visually, maybe under an electron microscope, that distinguishes ferroptosis?
Yes, ultrastructurally, ferroptotic cells show some characteristic changes, particularly in their mitochondria. They often display smaller than normal mitochondria with reduced or vanished cristae, the internal folds, and sometimes a ruptured outer mitochondrial membrane. These mitochondrial features can help distinguish it from, say, classical necrosis or necroptosis.
And this pathway, ferroptosis, is relatively new, right? Discovered around 2012?
Yes, it's a much more recent discovery compared to apoptosis or even necroptosis. And it's generated huge interest.
Why the excitement? Because it's linked to specific diseases? or because it seems druggable?
Both. Ferroptosis has been implicated in a growing list of conditions. It seems to play a role in cell death during stroke, traumatic brain injury, acute kidney injury, and interestingly, in neurodegenerative diseases like Alzheimer's and Huntington's.
Okay, disease relevance and druggability.
Absolutely. Because it hinges on specific factors, iron levels and GPX4 activity, it's potentially highly modifiable. You can induce ferroptosis, for instance, with drugs that inhibit GPX4 or deplete glutathione, which might be useful for killing certain types of cancer cells that are resistant to other therapies.
Ah, turn the pathway on to kill cancer.
Right. Conversely, you might want to prevent ferroptosis in conditions like stroke or kidney injury. This could potentially be done using iron chelators, drugs that bind up free iron, or compounds that boost GPX4 activity or provide alternative antioxidants. It's a very active area of therapeutic research.
So ferroptosis, iron-dependent, GPX4 regulated, lipid peroxidation-driven membrane failure. Okay. This really changes the whole picture of necrosis. It's not just one thing.
Not at all. We've gone from viewing necrosis as simple accidental collapse to understand the spectrum from classical, purely accidental necrosis triggered by massive injury to these highly regulated, programmed forms like necroptosis, pyroptosis, and ferroptosis, each with unique triggers, mechanisms, and potential therapeutic vulnerabilities.
Okay, let's try to synthesize this deep dive then. We started with necrosis as the endpoint of irreversible cell injury, that point of no return.
Characterized fundamentally by that loss of mitochondrial function and, critically, the profound disruption of cell membranes, especially the plasma and lysosomal membranes.
Leading to cell swelling, enzymatic self-digestion, and that crucial leakage of intracellular contents.
Which act as damps at damage-associated molecular patterns like ATP and uric acid, triggering a robust inflammatory response. Necrosis always causes inflammation.
And that very leakage is the key to clinical diagnosis for many conditions. Measuring tissue-specific proteins like cardiac troponin in the blood tells us necrosis has occurred in that organ.
A direct biochemical signal of cell death.
We then looked at the microscopic signs, the increased eosinophilia, pinkness, the glassy or moth-eaten cytoplasm, and those definitive nuclear changes, karyolysis, fading, penosis, shrinking, and karyarchically fragmenting.
The visual evidence of cellular demise.
And we explored how widespread necrosis manifests in tissues as distinct patterns: coagulative, structure-preserved, ischemia; liquefactive, liquid pus, infections of brain; gangrenous, limb ischemia plus sullen infection; caseous, cheesy TB, fat, chalky soaks, pancreatitis; and fibrinoid, pink vessels, immune reactions.
Each pattern offering diagnostic clues based on the gross and microscopic appearance.
But then we hit the paradigm shift. The discovery that not all necrosis is accidental.
Right. The regulated forms. Necroptosis, the programmed necrosis, driven by RIPK13 kinases, activating MLKL to rupture membranes when caspases are blocked.
Pyroptosis, the fiery death, triggered by inflammasomes activating caspase-1, which releases IL-1 and uses gasdermin D to lyse infected cells.
The ferroptosis, the iron-dependent death caused by overwhelming lipid peroxidation when the GPX4 antioxidant system fails.
Understanding these specific molecular pathways, RIPK, MLKL, inflammasome, caspase-1, gasdermin D, GPX4, iron metabolism, it completely changes how we think about intervening.
Absolutely. It moves us beyond just dealing with the aftermath of necrosis, inflammation, debris cleanup, towards potentially preventing or selectively inducing specific types of cell death by targeting these molecular switches.
Which brings us to the real significance for you, the listener. Why does distinguishing between accidental necrosis and these regulated forms truly matter?
It matters because it offers hope for treating conditions previously thought to be just unavoidable tissue loss. If cell death following, say, a stroke isn't just passive collapse, but an active, programmed necroptotic process, maybe we can block that program.
So consider this as a final thought, something to mull over. Imagine a patient having a heart attack. We rush them to the hospital, open the blocked artery, restore blood flow. That's crucial.
Essential.
But we now know that the reperfusion itself can trigger necroptosis, killing cells that might have otherwise survived. What if, in the future, alongside opening the artery, we could administer a drug, a necrostatin, that specifically blocks RIPK1 or RIPK3 activation?
Could we significantly reduce the final size of the heart muscle damage? Could we preserve more function, improve long-term outcomes after heart attacks, strokes, organ transplants, or other ischemic injuries?
By specifically targeting and switching off that programmed chaos, could we rescue tissue that today is simply lost forever? that ability to selectively control not just if a cell dies, but how it dies.
That really feels like the next frontier in treating a vast range of human diseases. Understanding and manipulating these death pathways is where so much exciting research is heading.